And there is one really lucky guy here who always has his slides ready. And so Florian Markowetz, who is part of the CONTRA, so he's going to be an exception that person from CONTRA is speaking on this workshop. So he's going to keep us entertained until lunch.
Good. Thank you very much. I was told to be quick. So I'll present 1.5 projects about ovarian cancer. No introduction, because by now you should have seen the slides on tumor evolution way too often. My lab does all kinds of things. We have never been accused of focus. What I present is mostly team blue, the evolution bit. You see Michael, who's sitting over there, and that's Lydia. She's also an ITN student. We do quite some imaging where we try to understand the tumor tissue surrounding all the cancer cells, because most people in this room are very reductionist. They just treat cancers as bags of cancer cells and count mutations. But there's tons of other stuff around that, and you have to actually look at it and use images to understand that. And I have a part of the lab which is actually a wet lab. We try to understand nuclear receptors, we do perturbation experiments and reconstruct little pathways. Now, this presentation is all team blue. It's led by Geoff McIntyre and is being continued by Michael. The question that Jeff asked is not about phylogenetic trees. It's a basic question of how do we get the mutations we see in the genome? What are the mutational forces, the mutational processes that shape the genomes that we observe and from which we infer phylogenies and little trees? We did this in collaboration with James Brenton, who's also at my institute in Cambridge, and this will all be, the first part will all be high-grade serous ovarian cancer. Now there's different types of cancer, some of them have lots of SNVs. Ovarian cancer is all copy number changes. So it's, that's kind of the defining feature, and that is what complicates all our analyses. The idea, what people have tried in ovarian cancer is to take a gene-based view, where they say, do we find recurrent changes in the genomes? Maybe there's this one region that's always amplified or always deleted. And that doesn't work. Very little has been found in ovarian cancer. These genomes are completely messed up. So rather than a view that looks at individual loci in the genome, we take a very global view. We are looking at what shapes the global distribution, the global patterns of copy number changes that we see. And this is motivated by the work that has been done on mutational signatures on SNVs, for example, by David and Serena Nixinal and all of this, all from the Mike Stratton school of thought. The idea behind this is the following. What we observe in a tumor, they call it here, I can't even read my own slides, a mutational portrait. So you see a certain set of mutations in the tumor. Now, how did it get there? Well, it got there because during tumor development, there were different mutational processes like maybe aging or smoking or UV light happening at different times. And each of these processes was contributing a certain set of mutations. That's these ones. That's the so-called signatures. So you identify a process by a certain set of mutations, and what you see in the end is the sum, the weighted sum, because maybe you got very little light, but you were a heavy smoker, so you get a different contribution of these two signatures, or maybe somebody else is a heavy smoker, but is also always out in the sun, so he gets both. So it's a weighted sum of the contribution of these individual processes. So the goal of signature analysis is take this final mutational portrait and deconvolute it in to the signatures and their individual so-called exposures. So they have done this for SNVs. You see, SNVs, it's so easy. They could do it at the Sanger. Whereas what we do is a much harder thing. We look at copy number. So we have a copy number, and the goal is exactly the same. We take the complete mess that an ovarian cancer genome is, and we try to deconvolute that into individual processes that shape copy number. One could be whole genome duplication. Another could be chromophripsis, and there could be many others. So the goal is, you give me one of those, And I tell you, well, it's a bit of copy number, a bit of whole genome duplication, plus 50% of chromothripsis, plus 30% of this other thing. Okay, that's the goal. How do we do this? The first step in all signature analyses, be it SNV or copy number or structure variance or whatever is, to take your genomes and translate them into numbers. So you map your genomes into some count space. So we start with a whole bunch of segmented genomes. We have absolute copy number for all these segments, and that's a story in itself, how you get there. In our case, we derived that from our cohort, we derived that from shallow whole genome sequencing. Shallow means 0.1x average across the genome, so it's very shallow. So each of these profiles cost about 50 pounds, so that's not too expensive. We take many of them. Our cohort is about 120. And we characterize them using 6 fundamental features of copy number. And by fundamental, I mean really basic things like what is the copy number of the segments? So if you have many segments, you will get a distribution of copy number along the genome. There will be some segments with copy number 4, some segments with copy number 3, and you plot this distribution, it would look like something like this. In this example, lots around copy number 4, but there's also some very highly amplified regions. Another basic feature is what is the size of the jumps between consecutive segments? Again, if you do this across the genome, you get a distribution. What is the length of the segments? Some are long, some are short. How many breakpoints do you see per chromosome arm? How many breakpoints do you see per genome? So it's pretty basic stuff. For each of these 6 features, you get one distribution. And then we use mixture models to find out which are the components of these distributions. And the number of components will differ for each of these 6 distributions. So in the first example, for the breakpoint count for 10 megabases, it turns out this distribution is made up of 3 different components, and you use some statistical techniques and then some model selection to come up with these numbers. So this is 3 components, whereas the copy number distribution is 8 components, and for example, segment size is 10. We derive these numbers by using the data from the whole cohort. So these, in sum, 36 components describe the way copy number looks like in our whole data. Now, if you come in with one particular genome, we just look up for each event that you see to which of these components does it belong. Is it a short one? Then it will be in the short segment size component. Is it a tall amplification? It will be in the one of the far out components for copy number. And we just count for each genome how often we see each of these 36 components represented in the particular genome that you're interested in. And this is a mapping that takes each genome and represents it by 36 counts. One count for each of these components. If you do this for, yes please. Yes. 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 So they evolved. They were partly because they're basic, partly because the distributions actually we can work with them. For example, we had one which was the distance from the centromere, because we thought we wanted to know where the breakpoints are they close to the telomeres or not. And it just turned out that when we worked with it, this distribution somehow looked funny, and in all our methods it behaved funnily. So we threw it out and put another one in, which is the breakpoint count per chromosome arm, which it turns out describes similar features in the data. So this is ongoing. So one of the ongoing things is fine-tuning these distributions, fine-tuning the mixture models, finding out if there's other features we could use. It's similar to what they do with SNVs, because there at some point somebody came up and said, oh, we could look at pairs. Somebody else said, oh, we could look at the 2 pairs to the left, 2 pairs to the right. So it's a whole evolving field. You need to figure out what makes sense. Loss of heterozygosity is one of the features we'll definitely include in the next version of this. So there's nothing allele specific in here so far. This is all total copy number. So this is evolving. But this is what we got so far. 6 features, pretty basic. Now, if you do this, if you take a cohort, and you represent each sample by 36 counts, you get a matrix like this. Our cohort is called BritRock because we did it in Britain and it rocks. 117 tumors. and we have the 36 components, and the colors correspond to the different counts we get. Now, we thought we don't want to burden the reviewers of our paper with too many new things, so we just use non-negative matrix factorization like everybody else on this matrix. If you do NMF, you decompose the matrix into two matrices. One of them has the number of tumors in one axis and a certain magic number of copy number signatures. And the second one has the number of components and the same number of copy number signatures. You use model selection again to find out how many signatures do you need to describe your data well. And there's all kinds of packages and approaches out there which work okay-ish. And by using the output of these algorithms and just eyeballing it, we came up with 7. So we are now claiming that ovarian cancer is shaped by 7 distributions. Here, in this one, you see how active each of the 7 is, and here you see which features contribute. Our signatures are very reproducible. So when we did it, this is our shallow whole genome sequencing. We did the pan-cancer ovarian data set, which was whole genome sequencing, and the TCGA SNP data, and look at these matrices. I mean, they look pretty much the same. Also, on the way, the reviewers and people like Wei told us that our method was not really good. So the mutational part is the one after review. What I initially proposed was something based on moments, which gives you pretty much the same results. You have a question? Sorry? Oh, sorry, they're the same. Sorry, they're synonym. We just use them. So we just have the segments, and at every point where you go from one segment to the next, that's we call a good point. We'll change this. Okay, so could still be very biased and very wrong, but at least no matter what data you give it, and with different algorithms, you get the same results. That makes me happy, because usually in genomics you get all kinds of random stuff when you change a little bit. Now we can start interpreting these signatures. So what you see here in the bar plot are the 36 features, like these 36 components of these different mixture distributions. And in signature 1, you see it's been characterized mostly by a small number of breakpoints, small number of breakpoints per chromosome arm, and large segment size. So it gives you an idea how it looks. Now what we did is we took the exposures of these signatures and correlated them with all kinds of other information that we had about the samples we used. For example, we knew the age at diagnosis, we used whether there were amplification-associated foldback inversions, we knew how long the telomeres were, we knew what the SNV signature said about the samples, we saw which genes were mutated, and we could group the individual gene mutations into pathways. And then we started associating the signature exposures to these additional features, and that's the matrix we got. And you see there's distinct patterns, which I show you in a second the results, which allow you to interpret them. So the last slide gave you an idea of how the genomes look like, and this gives you an idea of what the signatures are associated with. So if you take that together, you get a cheat sheet like this. This shows you how a signature looks. And the important components of signature 1, for example, are low number of breakpoints, 0 or 2 breakpoints per chromosome arm, large segment sizes. It's overall poor survival, higher in cases with NF1 mutations, correlated with amplification-based foldback inversions, anti-correlated with telomere length, and HRD SNV signature 3. So putting all of this together, we then say, well, this is the proposed mechanism is oncogenic, RAS-MAPK signaling, and telomere shortening leading to breakage fusion bridge events. So lots of manual work went in to kind of distill all this information down and come up with a story about all of the 7 signatures. So I show you the top 3. Signature 3, for example, is a BRCA-related HRD signature. For example, because it was highly correlated with the SNV HRD signature and with BRCA mutations. Now we have this for all of them. So we now have a catalog of 7 signatures plus associated stories of how they come about that characterize ovarian cancer. That is not so bad comparing if you look at the repertoire of SNV signatures, which is on bioRxiv. They have about 60, and half of them are UV light or they don't know what it is. So 7 out of 7 is good. And with some patients, we have actually interesting insights. So for example, we have a few patients which have germline BRCA mutations and somatic loss of heterozygosity. So in the clinic, you're like, okay, that's a BRCA case. And you see the HRD signature. That's the purple one, signature 3. It's present in all of them. That's good. But look at the other signatures. You see all kinds of other stuff. So if in the clinic you see that the BRCA patient goes one route or the other, we claim this is most likely because there's all these other mutational stuff. For example, look, the green one, number 5, really high here, very little there. That difference, we think, on what the patient does. And now using our methodology, we can quantify that in each patient. Now we did this for all the patients we had. So our cohort, TCGA, all kinds of stuff. I think it's about 500 that we have here. We ranked them by a Cox proportional hazard model according to the risk of death, and we computed signatures, which are the stacked bar plots. So each of these bars tells you what is the percentage of each of the signatures for this particular patient. And you see patterns. People who had a very high risk of death seem to have the green signature a lot, whereas on this other end, maybe, well, the brownish one seems to go up a little bit. So if you smooth the signature exposures in this plot, you get these lines. And that is, I mean, there's the survivor models, there are linear models. In a way, this reflects the coefficient of each signature in a survival model. And you see some of them go down, some of them are pretty flat, some of them go up. So that tells us a little bit of how they are associated with survival. We also tried unsupervised clustering on this matrix to identify subtypes of ovarian cancer, and we came up with 3. One of them is just the left end. So this is just, do you have signature 1? One of them, the right one, the red one, is just the right end, which is you have HRD. And the blue one is just all over the place. And in a way, this recapitulates the prior knowledge on ovarian cancer. What people have described is either one end or the other end, and just said, and in the middle, it's a mess. Now, it is still a mess. But with our signatures, we can quantify what kind of continuous mix of different signatures this is, which is better than just using dichotomies and saying it's either one or the other, and one of these classes is really huge and very messy. So we are now claiming there are no subtypes in ovarian cancer. It's a continuum of genomes. Do you want me to stop? No, no. Yeah.
So you can, of course, always model survival with something, right? And it will have different beta estimated coefficients, right? The question is how much variance do you explain when you use, I mean, how good of a survival model is it when you use these signatures as features? How much survival do they actually explain?
So yeah, so the question was, I mean, obviously you get, you always get some parameter betas, and they get, honestly, I don't know off the top of my head. Being overly, being a bit full of myself, I say we do really well. Right, I guess so, yeah. We need to look this up. So, what are the problems, the limitations so far? We did this in ovarian cancer, so we need to see what are mutational signatures in other tissues. So we currently do pan-cancer stuff where we just see if we apply this across 12,000 TCGA samples, what do we get? We saw a mix of signatures. If you're a mean person, you could say, well, I know why this is, because we all discuss different clones in the tumor, so maybe the mix of signatures is just the same as the mix of clones. So we actually have to look into single cells to understand which of these signatures co-occur. We used NMF mostly because we were lazy. It's very prone to outliers. It's not my favorite method, so there must be something better out there. In particular, what we are not reporting is any uncertainty on the signatures or the exposures, and ideally we would have that. I mean, that would take care of lots of these signatures, I think, which almost look the same if you could just put some uncertainty on top of them and see how whether they're really different. I mean, we don't have that, the other people do. So you find these signatures in the tumor. Now in the end we want to do something about patients in the clinic, so we actually have to find out which of them are historic of these mutational processes and they're just done, and which of them are still active. Because if I have a drug against something that happened 10 years ago, maybe that's not so interesting. If it's something that still happens in the patient, then I want to drug it. And what we also don't know yet is how we actually use that to make clinical decisions. The people who have subtypes have it easier in a way because they just have this clear-cut subtypes and we have this mess. So we somehow need to boil down our signature exposures into clinical decisions. And that's what we currently think about. Now, Michael came into the group 4 weeks ago. Yes, please.
Thank you. I was just wondering if you've considered using both mutations and copy number at the same time, rather than sort of correlating them afterwards.
So question, yes, we did. We're not the only one. Saurabh has a paper on this, so he was a bit quicker than we were. We have some ideas, but we never had a clear result. So we were thinking actually of just integrating all the different mutations together in some hypersignatures, right? But they're somehow very harder to explain than what we have here. So currently we're stuck with copy number. We've just looking at one particular feature of a genome at the time. Almost there. So Michael came and he took on the single cell project. So his goal is to quantify these mutational signatures now in single cell copy number profiles. And first steps are, do single cell genomes actually look the same as the bulk genomes? Are there any major differences? And so we have from the same sample, we have bulk and we have single cell, and depending on how big the bins are, these profiles look smoother or not so smooth. And when you eyeball them, amplification here, amplification there, amplification here, amplification there. Depending on how you massage the data, this can actually look pretty good. Michael, what type of single cell data is that? He said it's James Brenton's. Yes, I'll ask James what yeah, we need to look at, yeah. Hey, we started this 2 weeks ago or so. So the main challenge in here, in this game is, you need to transform your copy number into absolute copy number. So instead of just saying more reads, less reads, you need to know these exactly 3 copies, exactly 4 copies, which the way we see it is a scaling problem. So you observe after you segment, for example, we use circular binary segmentation, you observe all these different segments at different heights, and you need to infer a scaling parameter that maps these histograms that you observe to particular absolute copy numbers. And that's, for example, what absolute does for bulk, and we have different challenges here in single cell. We use the similar ideas. We use Gaussian mixture models with a scaling factor to do that. In real data, I mean, we drew them by hand, right? So they never looked that nice. In real data, these histograms look like this, so the mixture modeling is a bit more challenging. So what we currently work on is mixture models of these histograms. We want to do joint segmentation and scaling instead of doing it in one step after the other. And we would like to combine single cell plus bulk sequencing, because maybe the single cells can just guide but we have more read depth in the bulk, or if we have the data, we want to combine it anyhow. Good. With that, I thank all kinds of people. So Jeff was pretty much leading this. This is pretty much all of his work. If you have faculty positions at your institution, send him an email. Michael did a lot of stuff on single cells. And I hire if you're interested in imaging plus genomics. Thank you very much.
Okay, I guess everyone is really hungry, but that was an interesting talk. You can have one question. Okay, yeah, the usual suspect.
Florian, I'm just wondering, in this, when you used the moments, did you try using the unnormalized moments? Using the normalized moments was obviously a stupid thing to do, but did you try using the unnormalized moments?
Nobody gave me I think we did. It didn't really change much. Honestly, with this here, we always get the same result. I don't know. Yet, right? It was stupid. It was my idea. It was not Jeff's. So using the moments was my particular contribution to that paper. It's good we got rid of that. Yeah. Lunch? No. Ben.
So great. You talked a little bit about the feature selection in response to Nuri's question, but I wonder how correlated the features are as you sort of went through this process, because it seemed like after the signatures, you were seeing correlations that seemed to have make sense and be obvious kind of post hoc. So, you know, were those already in the features themselves? The features are highly correlated, so they come out in the same signature, or?
So these are two different correlations. One is between our 6 features, and there are correlations. I would like to get rid of those. So feature design is on the to-do list. But at this, so we were cheating. I mean, I always make fun of the SNV people who have such a hard time finding mechanisms.
Yeah, but theirs is actually much simpler because the features are not as correlated because you're looking at a single position.
Right.
So you change the window size a bit, but it's not...
We are cheating a little bit, and it's not cheating. We put lots of prior information in, so we know a lot of ovarian cancer genomes. So these features, they are correlating all of this, but we're chosen in a way to reflect what we knew would be in the data. So we knew there is foldback inversion. So of course we chose features that can find that again. So we handcrafted it for this particular example. And then when we do it genome pan-cancer-wide, let's see what happens. Thank you.
Okay, thank you very much.